I don't have to tell this audience that glycans are very important in all cells, and the several components, several glycoconjugates are make major part of this of every cell. And I also don't have to tell you, because this audience knows, the many factors interfere with the glycome of a cell. In fact, of course, the expression of the glycosyl transferases, and there are more than 200 genes codifying different families of glycosyl transferases, which have cell and tissue expression specificity. But there are other factors that can interfere with the glycome of a cell, which include molecular chaperones, donor substrate concentration and availability, acceptor substrate availability and concentration, and also glycosidases, endogenous and from external microbiome, for example. So, and this is very controlled, as you all know, at the cell and tissue and organism level. So I'm going a little bit into the understanding the functional aspects of the glycome in cells, including in cancer. And for example, in the normal gastrointestinal tissue, this is highly regulated. This is the gastric mucosa, in which in the upper part of the foveolar superficial epithelium, we have, for example, H-type 1 glycan, Lewis A biosynthesis, as well as Lewis B. And also, between normal individuals, we are polymorphic for several of these genes. For example, for the FUT2 enzyme that does just this link, according to the population, we will have major polymorphisms, and therefore we will have in normal mucosa or some individuals who will not have this glycan biosynthesis. And why is this important? Well, because this, in the context of a given tissue, in this case in gastric mucosa, these glycans function, as you know, as ligand for microorganisms. For example, the highly adapted Helicobacter pylori is a major bacteria that colonize happily the normal gastric mucosa of individuals, generating a inflammation that can cause gastritis, of course, and can cause further disease development. But in the gastric carcinogenic process, so before there is a tumor, there are several alterations that can occur in the tissue. For example, in this mucosa, it can appear a lesion that pathologists recognize as intestinal metaplasia, which, if you look just by this simple analysis with monoclonal antibodies, completely change the expression of the glycoproteome and the glycome. So basically, they switch the expression of the key mucins expressed in the gastric mucosa, in the green labeling, and express a intestinal mucin, MUC2 in this case. And together with this, a lot of glycosylation changes appearing of this truncated O-glycan, highly abundant in these mucins. So when we go for at the cancer level, many of these alterations are present, and several of them are very well documented. And yes, and of course, cancer cells, they do not invent things. They use what is advantageous and possible to do in biological terms. So basically, pathways of biosynthesis will be activated, and if they confer biological advantage, they will be kept in the tumor. So when we analyze a tumor removed from a patient after 1 year of development of 2 or more of the tumor, we analyze in the picture of that day of the tumor. So it's what has conferred advantage for the tumor is there. Some of these glycans, as already has been nicely presented today, are approved markers used in the clinic for different types of cancer. Unfortunately, as also has been mentioned, they have very strong limitations in terms of diagnostic. They are basically used for monitoring the patient after diagnosis, so monitoring the recurrence of disease and so on. But this does not function only as markers, as biomarkers. Glycans are important because they play key functional roles in the tumor context. For example, they can different alterations in proteins involving adhesion molecules like E-cadherin and integrins are affecting the mechanism of invasion of the tumor cell. And as I hope I can show you a little bit later, there is some alterations of receptor tyrosine kinase that are critical for the oncogenic signaling that is going on in the cell. And also, of course, the metastasization process in which glycans also play a critical role. So the first that I wanted to mention is the siALTN, because it's a very highly expressed tumor marker that is expressed not only in this case in gastric carcinoma, but in many tumors is highly expressed, so it's somehow selected in these tumors. Actually, we know in some cases how it is selected. And at that time, we wanted to understand what were the proteins carriers of this siALTN. So using the strategy developed by Catherine Steenstoff in Clausen's lab some years ago, Diana Campos generated a knockout of the cosmic mutant, cosmic molecular chaperone of this step of the biosynthesis of all glycans. So the protein remain just with the GalNAc. All the proteins in that cell will have GalNAc, and because the cell has ST6 GalNAc1, it will produce siALTN. So then, using this strategy, the peptides will, the protein will only have this shorter O glycan, either TN or siALTN. The glycopeptides could be generated by digestion and removal of the sialic acid, and then a long lectin affinity chromatography followed by mass spectrometry and protein identification. So, and this allowed us to define all the glycoproteins and all glycosides in those glycoproteins in this gastric cancer cell. And some of the proteins that were identified include CD40, were very known for the cancer field molecules like CD44, which were present not only in the cell models but also in the cancer sample from the patients. And CD44, just a little bit of the understanding of what the role it was playing. CD44 is a major co-receptor functioning with interacting with receptor tyrosine kinases, which induce oncogenic signaling. It also is a receptor of hyaluronic acid, which is very important also in the cancer biology terms. And these O-glycosylation were identified in different sites of the CD44, including in some variable regions due to splice variation of the CD44, which include V6 region. So which is I don't have here for the sake of time, but it's a region that is highly this form is highly expressed in cancers. And these models knock out for either the simple cell, knock out for the cosmic, but also overexpression of ST6 GalNAc1, or even having a shorter O-glycan by other types of glycosyl transferase manipulation, change the interaction with hyaluronic acid. Moreover, if we look at the cells in terms of expression of this in the three models using this proximity ligation assay, in which on one side we detect the V6 variant with a monoclonal antibody to this variant of the CD44, and to the others detecting a receptor tyrosine kinase, we saw increased signaling in all the the different independent models. Moreover, we saw increased activation also associated with the V6 variant. And in the tumor tissue, we could also apply this, I hope you can see the signal there in the back. We can see the signal of the detection of the CD44V6 and the shorter O glycan. We also detect this co-localization with increased activation of the receptor tyrosine kinase. I don't know how am I on time, but I will jump for the last part, maybe, because I don't think I have time. So the last part I wanted to show you is that, as I mentioned, cancer is highly heterogeneous. And, for example, in the case of gastric cancer, we have 4 clear molecular subtypes, which we can identify based on genomic and epigenetic alterations. And these are in biological terms, they are different. And also they have different targets. And in fact, some of them, in the case of gastric cancer, some of them can be, there is one target therapy approved, a single one, targeting the HER2 or CRB2 protein, which is expressed in a subgroup of patients. So these patients can be treated with an antibody targeting that protein, namely trastuzumab, the famous trastuzumab. So trastuzumab is an antibody that functions in different ways, disrupt ligand heterodimers, receptor internalization, induction and degradation, inhibit signaling, and also induce ADCC. So the interest in that protein started a few years ago when we observed that, when we digest the protein with PNGSF, there was a clear shift in the electrophoretic behavior of the protein, and also some glycans were disappearing based on antibody and lectin analysis. And actually, the interest got even further when we analyzed the tissue, the cancer tissue of the patients, and saw that some tumors within the same stage within the same stage of the disease, express particularly glycosylation features. And actually the expression, the tumor having this expression or not having this expression, showed a different biological behavior. So the tumor had a different prognosis within the same stage of the disease, a diagnosis. So we decided to go in depth, and we generated different clones, knockout for the STCs-Gal1, the enzyme that does this terminal alpha-2,6 cellulation, different independent clones by CRISPR-Cas9. I don't have time to go too much into detail, but I just wanted to show you that we, in collaboration with Manfred Hurer, we immunoprecipitated this glycoprotein, and Enrique Duarte performed an analysis of the glycoproteomic part of the protein, which you digest the protein into glycopeptides and analyze by LC-MS. And on the other protocol, perform a removal of the glycans and labeling chemically and do CMS for the glycan characterization. And so I don't have time to go into detail, but as expected, we have lower expression of alpha-2,6 glycans and glycans, and also increase of fucosylated and glycans, some also increase on alpha-2,3 cylillated and glycans. And the glycoproteomic analysis provided information about the site, the specific site where these were occurring in these cells. And if you put all the data together, we can have the map of the RB2, and some of these major alterations were occurring in the domain 4, which is the domain that trastuzumab recognized the protein, the therapeutic antibody recognizes. And so we perform a series of in vitro analysis with these cells and the clones, and show that there were major, this glycosylation change we're inducing major modification in terms of response to trastuzumab. And this is an assay using a dose-dependent response to the therapeutic antibody in vitro, and also changes in the levels of the receptor at the membrane of the tumor cells, and modification of the response to the therapeutic antibody in the presence of the ligand for the heterodimer of RB2 with the other members of the family. And I don't, yes, so with, this highlights one mechanism in which glycans can be modify the response of the cancer cell expressing the RB2 to the therapeutic antibody. So the vision is that in the future, we need to add, apart from that what we are using nowadays in the clinics, the genomics, transcriptomics, and so on, for the subtyping of the tumors, we need to add this major component, which is glycans, to better stratify patients. And with that, I finish, thanking all collaborators, and thank you very much for the attention.